Hi there. We'll just wait a little bit to see the attendees list sort of stabilize, but we will kick off as promptly as possible. Right. Good afternoon, everybody. And thank you all for signing up to this webinar related to the experimental medicine scheme that we run as part of DPFS. My name's Adam Babbs, and I'm the head of translation at the MRC. And I'm just going to quickly introduce a few slides before handing over to my colleagues, who I'll introduce a bit later on. So it's really great to see so much interest in this. This is obviously a scheme that we launched as a regular twice-a-year scheme in July 2020, and has been going from strength to strength. But we felt it was really important to sort of get out there into the community and sort of talk around what it is it's trying to do, and also ways that you can make the best possible application to this scheme. So if I can have the next slide, please, Helen. Yes. So today's webinar, we've got an hour sort of penciled aside. We're sort of aiming to be no more than sort of 25 minutes to talk through these various areas. I'm going to provide a quick overview just of MRC beforehand, before we go into the specifics here. And then that will give us plenty of time at the end for Q&A, which we'll do via the Q&A function. So if you want to start adding your questions as we go through the seminar and they spring to mind, we'll then go through and answer as many as possible. And if there are any that remain unanswered at the end of the webinar, then we'll try and put together a written document. We are also recording this today, and so there is the possibility that we'll be able to upload this and have it there as a resource that you can signpost to future people within your institution if they also wish to see it. So kind of in the next slide. So just a quick introduction to the team. We're a fairly compact team in terms of MRC. So there's myself, who's head of translation. So ultimately this falls within my budget line. And I'm supported really ably by two program managers, Helen Potts and Sophie Mountcastle, who are both on the call today and will present various elements. We also have a fantastic administration team that makes sure that all the applications come in and are properly processed and properly peer reviewed, and everything is sent out and everything is then in place so that we can make the highest quality decisions. And we have a standing panel where everybody has a 2+2 term on it, and we rotate the membership on a regular basis. The two key people on that panel are our chair and our deputy chair. So this is currently Professor Ruth Plummer, who's based up in Newcastle, and Dr. Fiona Watt, who is at Imperial College in London. Okay, the next slide, please. So since Patrick Chinnery took over as the executive chair of the MRC, this is the sort of mission that he's challenged us all with, which is that the MRC really should be looking to discover biological and pathological mechanisms understanding human disease. And in doing this, this will enable us to be earlier in our diagnosis of disease, come up with new ideas for treatments, and also to look at how we can actually prevent people from getting disease. Now, if you look at this, this all sort of fits with exactly what experimental medicine is trying to do. So for us, the experimental medicine scheme is one of the most important schemes that we could have in order to deliver against these high-level mission targets of understanding the pathological mechanisms of disease. We can have the next slide. But the experimental medicine isn't alone within this. So we're going to focus today really on the experimental medicine scheme. I'm not going to go into any details because my colleagues will talk around the specifics later on. But it has an £11 million a year budget at the moment, but it fits into a much broader set of schemes that we have for translational funding. And in this current year, we've actually had about £72 million that go into our various schemes. So if you get to the end of the seminar today and you think, actually, what I'm doing doesn't fit within experimental medicine, it may well fit into one of these other schemes. So there are plenty of opportunities to be looking to translate the high quality fundamental science that is done within the UK. And we're more than happy to talk about any of these other schemes outside of this webinar at a later date. I'll go on to the next slide. So I'm going to hand over now to Sophie Mountcastle, who's going to talk you through the elements of the panel remit.
Great, thanks, Adam. So, so experimental medicine's a broad term that MRC uses to refer to investigations undertaken in humans to identify mechanisms of pathophysiology or disease. And so the purpose of the experimental medicine study so your project, when you apply to the experimental medicine panel, must focus on a mechanistic hypothesis and include an experimental intervention or challenge in humans. And we're looking for projects that can clearly articulate a gap in understanding of human pathophysiology and have a clear path to clinical impact. And so projects that are successful through experimental medicine will produce new mechanistic insights, and including those that may identify opportunities to modify disease pathways or enable the future development of novel therapeutic or diagnostic approaches. Next slide, please, Helen. So we welcome applications in all disease areas and all types of interventions. So we funded experimental medicine research projects across the whole span of human health and disease. For example, studies focusing on cardiovascular disease, cancer, neurology, and infectious disease, but not limited to those. And interventions may include pharmacological, immunological, physiological, psychological, or infectious modalities. But again, that's not limited to those. We have an £11 million annual budget, as Adam mentioned, and there's no limit on the duration or cost of the project that you can submit. So these really aren't standard 3-year research proposals. They should be kind of based around the time and the cost needed to deliver the project aims. So we see projects that are much shorter and longer than that standard kind of 3 years that you might typically see. We fund awards from sort of all the way from 200k up to over 2 million. So there's and regularly fund projects that are over £1 million. And so what we're really expecting is that your project is based on the scientific need and the project deliverables when you kind of ask for the duration and cost of your proposal. Next slide, please, Helen. So this slide's really sort of trying to explain the different types of mechanistic hypotheses that we see applied in applications to experimental medicine panel. So we see applications that cover things such as mechanism of interaction, mechanism of disease, mechanism of action of the intervention, and confirmation of tractable disease pathways. And you can see from the slide, the graph shows that we see applications across all of these types of mechanistic hypothesis, and we've awarded grants that address all of these different types of mechanistic hypothesis. So these are all kind of eligible within the remit of the scheme. And I've got on this slide some examples of eligible and ineligible activities, so I'll go into a bit more detail about those. So examples of eligible activities would be the use of drugs, other interventions, or measures with an established safety profile in new settings or conditions. So, for example, repurposing drugs as a tool to probe disease mechanism, or the use of novel readouts or technologies related to early evaluation of pathogenic mechanism. We do permit the inclusion of limited and well-justified elements of non-human work, but for this panel, the focus of the project should always be on the human participants, and any non-human work should be used to inform and improve the experimental human part of the study. We also permit limited hypothesis-driven retrospective sample analysis at the start of the project, which may be needed to improve the design of the experimental study. And again, your application should clearly detail what data you need from this analysis in order to deliver the experimental medicine study. We also permit prospective nested studies within a larger cohort trial, provided that they add value and are exploring disease mechanism, and that they're testing a novel hypothesis and addressing a different question to the main study. So kind of in summary for what we would consider eligible, your proposal, your study design should aim to address the mechanistic question, and you need to provide a strong justification for why you've chosen those kind of experimental systems to probe and test the presented hypothesis. In terms of ineligible activities, so there's a few here on the slide, anything that's sort of characterization, aiming to elucidate disease etiology, biomarker discovery, projects where the intervention or challenges focused on animal work, or preclinical model development and validation are not within remit for experimental medicine. These areas are covered by the research boards. High throughput screening approaches to target validation we wouldn't expect to see, because we would expect your study to be kind of targeted to a specific hypothesis. Development of and evaluation of novel therapeutics, diagnostics or devices would be covered by MRC's DPFS scheme. Clinical efficacy trials are supported by NIHR. So these studies aren't based on clinical efficacy, it's all focused on that mechanism. And we wouldn't fund an observational study that doesn't involve an experimental challenge or intervention in humans. So I guess the main takeaway is we really encourage applicants to contact the experimental medicine team in advance of your submission to discuss kind of whether your proposal is suitable for the scheme and talk about kind of remit suitability before applying. But hopefully that adds some clarification on kind of the types of projects that we're expecting and what would be considered in and out of remit. Thanks, Helen. Next slide. So next I'm going to talk a bit more about the application process itself and what to expect from the scheme. So we have a two-stage decision process in experimental medicine. Stage 1, applications are reviewed just by the panel. And if invited forward to stage 2, your application will also undergo external peer review, and then a funding decision will be made by the panel at the panel meeting. As Adam said, we have 2 panel meetings a year in June and December, and the corresponding stage 1 deadlines fall in October and April each year. I think on the slide there's some success rates, Helen, I don't know. Oh yeah, okay, great. So yeah, that's just our current success rates for getting through each stage. After each stage, the panel will give you feedback on your application regardless of the decision. So hopefully we can help improve applications over time. Next slide, please, Helen. So at the stage 1 panel, the panel are really looking at to see whether the application fits a series of assessment criteria. So firstly, does the application fit the remit? Does it involve an experimental intervention or challenge in humans designed to explore disease mechanism? They'll also look at the rationale for the study and the potential impact it could have. So is there a clear justification for the hypothesis? Is there a gap that needs addressing? And is there a path to clinical impact? The panel will also look at the study design and statistical analysis plan and the approach to answer the mechanistic hypothesis, and also look at the go and no-go decision points in the study. And my colleague Helen will talk a bit more about milestones later in the webinar. And lastly, they'll look at kind of deliverability and feasibility. So is your project plan realistic? Does it offer good value for money? And when the panel are assessing value for money, the focus is on, justified resources. As I said, we're not looking for cheapest applications, we're looking for appropriately costed and resourced applications, and we consider there's no limit on what you can apply for. And finally, if the team have access to the necessary skills and resources to deliver the plan. So I'm not going to talk about stage 2 assessment criteria at this point for this webinar, but we do work with applicants when they're invited forward to stage 2 as well, on the feedback from the panel. There's 3 possible outcomes to a stage 1 panel decision. The first is that they think that the application meets all the assessment criteria and would like to invite forward a stage 2 application. You will still receive feedback from the panel at this point with any areas they think will need to be addressed at stage 2. The second outcome is that the panel might feel that the application doesn't meet the assessment criteria sufficiently and will decline to invite forward to a stage 2. Again, you'll still get feedback from the panel on the reason for that decision. And the third outcome is where the panel feel like it's got a potential in terms of the project, but they've got some questions around some of the assessment criteria, and they want to see a revised stage 1 application addressing any feedback that they have for the applicants. So you'll be invited to resubmit a stage 1 application. I think that's, well, next slide. Yeah, so now I'm going to hand over to Helen to talk a bit more about how to write a successful application.
Thank you, Sophie. Yeah, so now I'm going to discuss about some of our tips for writing a successful stage 1 experimental medicine application. So our first tip is to tailor your proposal to the audience. So as Sophie discussed, really at the stage 1, your proposal is going to be assessed by the panel. And so although all our panels are experts in experimental medicine, as you now know, the remit of this funding scheme is very broad. It's completely disease agnostic. So our panel may not be experts exactly within your field. So it's really important to tailor your proposal to this panel of general scientific experts. Next key thing to think about when writing your proposal is who are the project team, who are the key people you've got involved in your core team. And this project team must be sufficiently broad to cover all and support all aspects of your project, whether that's from including a project manager who's going to oversee the deliverables, whether or not that's including your various scientific leads as well as clinicians. As a clinical trial, any experimental medicine study will be required to have the appropriate statistical expertise or support. For instance, where appropriate, this could be the involvement of a clinical trials unit. It might be including a statistician as part of your core team, but really, what we need here is just to see that there's the appropriate statistical support within your study. Another key tip is to clearly state what is your mechanistic hypothesis and demonstrate how your proposed study design will test this hypothesis. And another key area is to carefully consider your project milestones. So I'm now just going to go into a bit more about experimental medicine awards and milestones. So in the MRC, all experimental medicine awards are milestone gated. And really, what does this mean? So our milestones represent these critical go, no-go decision-making points in your project, and they're really used to assess project progression. So kind of a key area that applicants typically fall down on is thinking of milestones very much linked to work packages, but really milestones sit outside of what the work packages might be for your project, and they really are superimposed on the project, and they really represent these go, no-go decision-making points. The key thing about milestones is they're never overlapping, they're always sequential in time and consecutive. And really, why do we ask you to gate your projects by milestones? And really, milestones enable the MRC to provide long-term commitments to inherently risky projects. As it's all experimental medicine projects or clinical trials, we know that there are inherent risks, and sometimes we might need to terminate a project early if it's not continuously progressing, it's not continuing to be successful. So really, when you're thinking about your milestones, it's really important to think about what are the key deliverables that I need to achieve to continuously progress my project along its project plan. And really, by gating each experimental medicine award by milestones, it really enables us to, as I say, provide that long-term commitment. And the way it does this is that the funding for each experimental medicine award is linked to each milestone. So within each milestone, we ask you to tell us how long it will take to reach that milestone and how much cost is associated for that milestone. And really, as you're working towards milestone 1, you'll really be able to access the costs that you've put aside for milestone 1. Once you reach that milestone 1, we ask you to write in a milestone 1 report. We assess whether or not you've met your success criteria for each milestone, whether or not you can progress to the next phase. And as you progress to the next phase, we're then able to release the next amount of funding towards your second milestone. So some key things to think about when defining your milestone success criteria is we encourage you to use this kind of SMART criteria, in which each milestone success criteria is defined by specific, measurable, achievably, relevant, and timely goals. So specific objectives mean that they should be really written in a clear and concise and understandable terms for each of your success criteria. Measurable means what are the measurable outcomes which allow us to determine whether or not the milestone has been delivered. Really ask you to set realistic and justifiable success criteria with a target value ranges, with it also acceptable cutoff values. That acceptable meaning what is the minimum threshold that you need to achieve in order to pass your milestone, and target being what's the ideal threshold in which you'd like to achieve. Achievable, this really means that we think of your milestone objectives, they should be really realistic, don't be too ambitious. Relevant, this means that your milestone success criteria really must focus on progressing the project towards the completion of the project plan. And timely also means that your milestone objectives should have a realistic time frame with an end date assigned to them. And these end dates really are what is the deadline? So which month do you need to have achieved your milestone by, not durations. So I'm just going to go through some examples about what makes a good milestone with the example of what would we think of as a poor milestone success criteria, what would we think of as a good milestone success criteria? So for instance, we might see a milestone criteria of complete participant recruitment, and this would particularly be a poor milestone criteria as it would be very vague. In contrast, a good milestone criteria, as you can see here, would be, I'm going to show you now how it would meet each of those SMART goals. So for example, a good milestone criteria would be recruit 50% of the study participants with a rate of 4 patients per month, an acceptable rate of 2 patients per month by month 12. So specific being that we're wanting to recruit this objective of at least 50% of all study participants have been recruited. Measurable, as we know how many patients we're expecting to recruit per month, as well as this achievable goal of what's our minimum acceptable target, acceptable value we want to achieve. Relevant being as we really want to know how the patient recruitment is progressing according to the project plan, and timely with this well-defined deadline by 12 months into the study, we want to see half of the participants being recruited. As another example of what would be a poor milestone, we might see a milestone in which where an applicant might not have understood that milestones really represent these critical go, no-go decision-making points. So in here, you can see that a poor milestone would be secure or necessary regulatory approvals. That would be a poor milestone as it's really vague in terms of what are the regulatory approvals needed. It's not specific. Additionally, this would be a poor milestone as the target value is to satisfy this by 6 months and the acceptable value is to satisfy this by 9 months. But really, the length of a milestone for both the target and acceptable value should be the same duration. So what would be a better version of this milestone would be to obtain approval from the Research Ethics Committee and Health Research Authority. So that's very specific. We know what approvals are needed, and ensure study sponsor approval and clinical setup is ready for recruitment within 9 months. So again, that's measurable, it's relevant, it's timely, it's well within an achievable time frame. So in addition to some of those common pitfalls we've just talked about in terms of milestones, I'll also just go through some general common pitfalls which we see where applicants typically fall down on in their application. So the first one might be insufficient justification of the clinical need. So as we have discussed this experimental medicine panel, remit is all about understanding the mechanisms of and the underpinning human disease. So really understanding what is the clinical need for us to get this increased mechanistic insight. Another common pitfall was when applicants may submit a project where it's really unclear how the study design will answer that mechanistic hypothesis. We typically might see a project where it's a really interesting and cool experimental system, but it's asking the wrong question, or equally that it's a really good mechanistic question, but the experimental study design is not well designed to answer that mechanistic question. Another thing we might see is that the team does not have all the expertise required to deliver, and the panel really will make an assessment about the project plan and as well as its execution. Another pitfall would be that the trial design and sample sizes and the experimental power are not fully justified. So really a key recommendation is when you're submitting your stage 1 application, it's really important to include a well-detailed statistical analysis plan and make it really clear how you've calculated any sample sizes or power size calculations to the level of detail that an external statistician can come and take your application and replicate any of those calculations. Another common pitfall is that the project doesn't seem feasible or deliverable, that the milestones are not measurable and they don't represent those critical go, no-go decision-making points within the project, that there might be a lack of consideration of any risk mitigation plans as well as patient safety. And a final common pitfall is that there's a lack of clarity on the roles of the project partners in their commitments. So some final tips when thinking about your application is if you're going to be working with any industrial partners, for instance, if they might be providing your pharmaceutical intervention at no cost, we have the MRC Industry Collaboration Framework. So at stage 1, we don't require this form to be filled out. That's only required for invited stage 2 projects. But at stage 1, we will really want to understand what is the relationship with any industrial partners that you might have in your project. So it's really important to think about this. I'd really recommend going to our web pages on the Industry Collaboration Framework if you have got an industrial partner for your project, and really understanding and laying out within your stage 1 application how will any IP be shared and managed within your project. This is just for any projects that are working with an industrial partner. I've also included some additional online resources that you can look at and refer to later, which talk about tips on grant writing. Gateway to Research is our online database of all previously funded UKRI research. So you can go on there and have a look at previously funded experimental medicine studies. So we have our Insight blog, which has additional articles on how to write applications and kind of common things we're looking for from a successful applicant. One of our key recommendations is to really contact us before applying. We'll be able to talk to you about your project, its fit to the panel, and kind of key things to consider when writing your application. And the final part of our webinar is I'm going to talk to you now about the new rollout of new investigator research grants to the experimental medicine panel. And really, we are starting to roll out these new investigator research grants to our experimental medicine because we know that there are existing barriers for early career researchers specifically entering into translational medicine and translational research. And this typically can be due to things like the inherent risks of translational projects, their extended timelines, the team science nature of translation means that these represent barriers specifically for researchers at that early career stage, as kind of the reward structures of academic science is often not well catered for within translation. And at the moment, although early career researchers are able to lead any experimental medicine projects, we don't have any specific funding mechanisms to support early career researchers. So really, to improve our support of these early career researchers performing experimental medicine studies, we're rolling out the new investigator research grants. New investigator research grants are a well-established funding mechanism that specifically support early career researchers and are currently offered in MRC's research boards. The new investigator research grants are really aimed at researchers who are capable of becoming independent principal investigators and who are now ready to take the next step towards that goal. New investigator research grants, also often referred to as NERGs, provide specific support for early career researchers by firstly providing a framework for research organisation commitment to those researchers transitioning to independence. And they provide that specific support for early career researchers as they provide a ranking uplift for NERG applicants if their applications are deemed fundable. And what that means is if we receive any NERG applications at stage 2, if all applications will be scored regardless of whether an applicant is led by a NERG or not. But if we are in a situation where we have a limited amount of budget and we have a few applications that received an 8 and we can't fund all the 8 applications which score an 8, those which are led by NERG applicants will go to the top of that pile and they will be prioritised for receiving funding. And really what's key to the NERG is this partnership between that new investigator, so between the researcher and their host organisation. So what does that mean in terms of the application process? So as we're currently rolling this out, at stage 1, so in this first round, there will be a new yes/no question to indicate whether or not you will be applying as a NERG. And at stage 2, so if you're invited to stage 2, there'll be 2 additional questions that only need to be answered by NERG applicants. And these questions really focus on eligibility of the project lead to be a NERG, as well as questions around the career development. So how will the NERG support the career development of the primary applicant, as well as what support is being provided by that research organisation to the NERG. So if you want to find out more about our MRC's new investigator research grants, I've also included this link. This is just to note that at this moment in time, this webpage does not currently have the updated information that shows that NERGs are now eligible for the experimental medicine funding scheme. And really, if you are thinking of applying as a NERG, so that would be if you are the project lead and you are eligible to be the NERG, you could apply underneath this framework. And we really encourage you to, again, reach out to us and we can discuss to you like the changes and what are the sorts of things you'll need to consider in your application. And with that, I'd just like to thank you. That brings us to the end of our webinar. In fact, I'd like to thank you all for taking time out of your busy day today to listen to us and learn more about the experimental medicine panel and our funding scheme. That I'm going to hand us back to Adam, who's going to lead our Q&A session.
Thank you very much, Helen and Sophie. So we've currently got 4 questions in the Q&A section, but feel free to keep adding them in there if you've got them on your mind. I'm just going to run through them in the order that they came in. So, Sophie, is the clinical trial unit involvement essential?
No, is the answer to that. The panel are really looking, as Helen said earlier, that you have the right resources and support available. So for your particular study, it might not need CTU support, but it just depends. It is dependent on what you're trying to do and what other members of the team you have. So it's not essential, but I think you just need to justify that you've got the right kind of support to be able to deliver the study.
Thank you very much, Sophie. So Helen, you just touched on this a little bit with the Gateway to Research element, but do you have a funded project list to see what has been supported in the past?
It's something that we're currently working on to update our webpage so we can have a better, easy to navigate list of links. We do publish previously funded applications available on our webpage, so it's something that we can circulate to all webinar attendees today, that list of previously funded links on our Gateway to Research.
Thanks, Helen. So Sophie, would a study using an agent in phase 2 development be eligible if the study was focusing on mechanistic understanding in the disease and not efficacy of the drug?
Yes, so I think the key question with that is that you can show that the intervention you're using or the drug you're using is safe to use and will have the effect that it's needed to probe the system. So you just need to provide enough supporting evidence that your intervention is safe. But if it's focused on a mechanistic question and it's not a clinical, it's not your sample size isn't calculated on clinical efficacy, then I think so.
Thank you, Sophie. So Helen, do we need to collect any clinical efficacy data before applying for this mechanistic grant?
Thanks, Adam. So very similar to as Sophie just mentioned, for what will be needed for any intervention that we use is it needs to have a well pre-established safety profile, but we don't need to have particular clinical efficacy for this trial. We will want to see data that your intervention is probing the mechanism as you say it is, as you predict it is doing, but we don't need that efficacy. We just need well-established safety.
Thank you. So I'm going to lump the next two questions together for you, Sophie. So what is the eligible criteria for a NERG? For example, is there a criteria on years of postdoc experience or age? And the second linked question is, what is the definition of an early career researcher?
So there isn't a criteria on years of postdoc experience or age because we understand that people have kind of varied career paths and may take time out. So we don't use kind of criteria like that to describe a new a NERG or an ECR, but really it's those people who are just sort of becoming leaders in their field, like leading grants, leading projects, they're just starting to step into taking on that PI role. I don't know, Helen, if you want to add anything to that.
Thanks, Sophie. We have again really detailed web pages which go into our eligibility criteria. As Sophie said, we don't have any specific durations of postdoc age or years. And the second phase of the question was, can you include PI salary costs in the application? Yes, so you can apply for up to 50% of your full-time equivalent within your NERG application. So that's to enable you to dedicate the time that you need to lead this project, as well as being able to combine your NERG project with other activities that you may be requiring, whether or not that's teaching or lecturing.
Thank you very much. I think we've answered 4 questions in one go there, so rattling through them. So back to you, Sophie. Are you allowed to include multiple interventions that all focus on the same therapeutic mechanism?
That's a good question. Yes, again, yeah, I think you'd be allowed to use multiple interventions as long as you've got this mechanistic hypothesis and can clearly articulate how those interventions will probe that question. So it's all comes back down to really to what's your hypothesis and how will your study design or your intervention probe the system to answer that hypothesis.
I'm going to skip over the next one and I'll come back to that maybe if we've got time later because it relates to DPFS. Helen, If we are working with an industrial partner, is there a ballpoint amount of IP you would expect the research institute to hold in comparison to the industrial partner?
Thanks, Adam. So if you're working with an industrial partner, we have two different ways in which you can apply to work with an industrial partner. One of them is under flexible IP considerations, and one of them is under gated IP considerations. And depending on which of these routes you go down really depends on whether or not your industrial partner wants exclusive rights to purchase the IP from your research organization at the end of your project. If you go down the flexible IP considerations, your industrial partner is expected to contribute at least 15% of the full economic cost of the overall project grant. And under fully flexible considerations, there's the opportunity for your industrial partner to purchase that IP at the end of the project, but they don't have that guaranteed pre-negotiated right to do so. So you'd be able to keep the IP either within your research organization or look to work with other industrial partners. If you go down the gated route, this gives your industrial partner that pre-negotiated and exclusive right to purchase the IP at the end of your project. But in that scenario, we really expect a 50% of the full economic cost contribution from your industrial partner. So there's no absolute threshold in terms of amount required to be contributed by your industrial partner. Also, just to make and clarify the point that any contribution from your industrial partner can either be cash or an in-kind contribution. That in-kind contribution could be things like staff time, access to resources and equipment and infrastructure, as well as providing potentially consumables free of cost. But there is no absolute threshold in terms of a money contribution. It really depends on the cost of your project, as that will really define that 15% and 50% contribution.
Sort of just add on to the points that Helen's made there as well. There are two categories where we actually ask you to define whether it's basic or applied. So applied applies mainly to our DPFS grants because that's where you're generating information that's specific to the product or prototype that's being tested. Within the experimental medicine grants, even though the drugs that are being used actually might be proprietarily owned composition of matter with a pharmaceutical company, they can be used as exemplars of something that targets a pathway. And if you are then going to publish that data and put it out into the wider community, that would still be construed as basic information because it's not necessarily generating data that's specific to that project. You're using that product or drug to actually identify mechanisms and pathway involvement. So then it would become basic in its nature. And that, if it's going to then be published, that intellectual property, which includes data, know-how, not just patents, is obviously put out into the public domain and can be used for anybody. So that makes things a bit simpler there. Sophie, next question. For the milestone gating, are they pass-fail or could the next stage of work be adapted on failure, e.g. if a hypothesis is not supported?
It's a good question. So Helen didn't have time, we didn't really have time to go into it on this webinar, but we have a process by which milestone objectives or timeframes can be adjusted because we know that with these types of studies, sometimes there's unforeseeable events that happen. So for example, with Helen's example she gave, if you have regulatory approvals by 9 months and site set up by 9 months, and then something happens that means that you're not going to meet your milestone, it won't mean that the project would automatically be failed and stopped. It just means that we'd have a conversation with you around mitigations and how much longer is needed and kind of the reason for that delay. So they are kind of, I guess, go, no go decision points, but they're not necessarily pass fail. It's more around if you feel like you can still meet the milestone, but you need more time, or you need additional staff, or you need to go to a different supplier. Those are all reasons we could consider kind of revising the milestone criteria. With the bit about hypothesis not being supported, I guess that might be a point to decide, if your mechanistic hypothesis and your intervention doesn't probe the system in the way that you anticipated it will. It might be a conversation about, what the next steps are for that study and whether or not, it's worth continuing and if it's not going to be able to be successful. So, but we'll always talk to award holders about that. It would never just be stopped just because you didn't meet a milestone, like straight off the bat. We'd always have a conversation with you about that.
Thanks. And to provide a real world example of that, we've had a DPFS project in the past where the results were exactly the opposite of what was predicted before we went into the study. And actually we stopped the clinical trial because it was causing harm rather than benefit. But what we did do is allow them to use the remainder of the money on the grant to actually try and understand why it had done exactly the opposite in a human as would have been predicted, so that we can get the most value out of that data, but also get that data out into the domain, into the public domain, because the rationale for doing it in the first place was rock solid because the panel supported it. So it's important that the rest of the community doesn't end up doing that experiment again, which can potentially cause harm in humans. So part of those, if it does fail, we will have that, as Sophie said, we'll have that conversation with you, because we could look to actually better understand why that result was in that way. Helen, can you clarify the degree of PPI required prior to stage 1?
Thanks, Adam. We don't have a absolute requirement, but we really encourage that there is that input of end users, and a panel will make an assessment about whether or not your study has really consulted with those end users and is meeting that kind of clinical need. So we'd really encourage things in terms of getting your PPI advisory group, having their input into the study design, whether or not that's also, you know, thinking about things like, is the study feasible? Will patients want to be recruited to this study because there's multiple site visits? Are there ways in which to make those site visits either shorter so you can come for just one site visit? Are there ways in which the time of them is needed? Are you proposing an intervention that would be very painful and people are not going to want to undergo multiple rounds of the same intervention? So we really encourage that within your project.
Thanks, Helen. Sophie, would a pilot study into the mechanism of action, efficacy and safety of a formula made from previously proven efficacious nutraceutical compounds be eligible for this funding call?
Based on the information in the question, probably not, because if it's not got an established safety profile, it sounds more like a product development. If you're formulating a new compound, then it might be more appropriate to take that elsewhere. I don't know what you think, Adam. It sounds from the question like if it's not got safety data, and even if it's previously efficacious, it depends on how the study's structured. It's quite difficult to answer based off that question.
I mean, to add to that, you know, if you have a nutraceutical, that would be considered challenging the human. So as long as it is a safe intervention of the human, and then the questions you're looking to answer are mechanistic, then it doesn't matter whether it's a nutraceutical or a therapeutic drug. If you actually want to take a nutraceutical and look to see whether it's efficacious in a disease setting, then that would fall under DPFS's remit, because that's them taking a product through the development cycle. Helen, you mentioned the importance of full statistical details. Is it advisable to upload a SAP? I'm just thinking of space in the stage 1 application. I'm not sure what a SAP is.
I assume SAP is like statistical analysis plan. Is
okay.
I'm going to guess what that acronym means. For the stage 1, we do know that the amount of space is limited in that approach section. What we really recommend is that there is enough detail around the kind of key calculations like your sample size and any power calculations. So it's really clear that the results you're going to get from your project are going to answer those key mechanistic questions. But we do understand that that space is limited. So there is always that balance between going into full amounts of detail versus being able to get across your point. So based on that, I recommend kind of focusing on those key areas, like your sample sizes and power. And
a related question, Sophie, around statistical support. Biostatistical support for specific endpoints are helpful, akin to those for a clinical trial, but some analysis may be related to multi-dimensional omic data that would be more the remit of a postdoc, for example, than a biostatistician. So do you want to comment on?
Yeah, that's absolutely true. And I think just echoing Helen, that you have the right people in place to answer the question and deliver the project. So yes, statistical support we think is really important because it's in humans. You know, your study design the number of participants to make sure that you get an outcome that answers the question is important. So we do recommend that you have statistical input in terms of the study design. and throughout the grant as well. But yes, for things like omics analyses, if it's in the remit of a postdoc's expertise, then absolutely that would make sense.
So Helen, how's the MRC experimental medicine different to NIHR's EME scheme?
Adam? So the NIHR's EME scheme is really looking a lot around efficacy and that evaluation. That's one of their key outcomes. In our scheme, we really expect all your studies to be focused on the mechanistic outcomes. You can have secondary endpoints which look at efficacy data, but we're really on the kind of discovery science in humans phase of of your project. So really thinking about making those mechanistic discoveries, whereas EME sits at kind of the later stages of translation.
Thanks, Helen. We will try and dig it out, but I'm pretty sure there's a resource where we compared experimental medicine, EME and DPFS to sort of talk through what they do differently. So we'll try and find the URL for that and add it to any information we send out afterwards. Sophie, is there any scope for an application to have 2 joint lead applicants? And if one applicant was a NERG, would the application be considered as a NERG application?
I don't think you need to have one primary applicant for UKRI grants, and I would query if there were co-applicants if the NERG is kind of stepping into that leadership role. So and sort of leading on a grant. So obviously you can have as many co-applicants and co-project leads as you need to deliver the project, but we'd always expect to have one primary application. And with the NERG team, it is about sort of indicating how that early career researcher is like stepping into that leadership role. So yeah, it needs to sort of fully justify that, and then the team around them to support them. I hope that answered the question. Thanks.
Helen, although all diseases are welcome, is part of the strategy to fund specific disease areas of unmet need?
Thanks, Adam. It is something that we have previously discussed with our panel members if there were any disease areas we wanted to make a strategic focus. What we actually think is really great about the scheme is that we don't have any strategic focuses of their disease. So all areas are welcome, and that really represents that there's a real breadth of disease across the kind of spectrum of human health. And that really will lead to great new insights. So no, we don't have any strategic areas at this moment in time.
And that answer is completely correct for the experimental medicine panel, but sometimes we do get money from government where we might use the experimental medicine panel as a mechanism to actually fund that specific fund of money. So there may be opportunities that sit for specific calls, but the experimental medicine panel per se is exactly as Helen said. Sophie, how do successful candidates articulate the effect of the experimental plans to economic growth?
So I think, is this related to Adam's introductory slides about MRC's mission? So I think the panel would probably more be interested in the kind of clinical unmet need to the question. I'm not sure that economic growth's been addressed as a key point in our applications unless I'm wrong. I don't think so. I think it's definitely more a focus on the clinical unmet need than the economic growth, and what the kind of downstream outcomes could be if the study was successful. What are the opportunities to then develop a new therapeutic or target a new pathway?
Yeah, I mean, that's exactly right. MRC has a general goal of increasing the health and wealth of the nation. And by increasing the health of the nation, invariably we increase the wealth of the nation because we become a more productive nation. So just increasing our fundamental understanding of disease will in itself probably spark economic growth. Helen, do these grants allow for costing in international collaborators?
Adam, yes, you can apply for costs for international collaborators for experimental medicine awards. We have a funding cap of that there's only a max of 30% of cost of the overall project grant can go internationally, but otherwise we really recognise that team science and international collaboration is key to some success. What you will be expected to demonstrate within your application is why you need this international expertise, why they're the most relevant person to help deliver the project. Yes, the international collaborators are welcome.
Can't remember who answered the original question. I'm going to go back to you, Sophie. So this is just a sort of follow-up to the question earlier around the assignment of intellectual property. So Let's just sort of clarify whether the research institute needs to hold 100% of the background IP, or whether a split where the percentage background IP held by the industrial partner is higher than the percentage of the research institute would still be eligible.
There's no criteria around background IP if a project partner is bringing background IP to the project, but it's more around the foreground IP and how that's dealt with. So kind of what the public money is paying for and how that then is distributed between the collaborators. So there's no, if you've got significant contribution of background IP from a project partner, that wouldn't necessarily mean that you can't apply through the ICF route.
Thanks, Sophie. Helen, in stage 1, is it appropriate to refer to any existing IP held by the applicants, e.g. patents?
Thanks, Adam. Yes, that would be appropriate to include that, just to demonstrate that you have the background IP to deliver on your project. It's not something that, again, the panel are making a decision on about whether or not you have any patents available. What the panel really want to see is what's the clinical need and what's the new mechanistic understanding that we're going to be delivered from your project.
Thanks. Sophie, it was mentioned that up to 50% of a salary for a NERG can be included. Is salary support limited to the PI, or could a percentage of a pre-doctoral fellow salary support also be included?
Yeah, it's not, the salary support's not limited to the PI for a NERG. You'd still cost your grant with the appropriate staff and the appropriate salary costs needed to deliver the project. It's just that the 50% salary is because we know that as career researchers establish their independence, they often have, perhaps sort of more focus on certain projects, and therefore that better institutional support if we can contribute a higher proportion of their salary than we might for an established group that have kind of a broader scope of projects and might use less of their time. So yeah, absolutely, you can cost co-leads and postdoc staff as needed to deliver the project.
Thanks, Sophie. There's one for you, Helen, and I don't know if you'll have actual statistics on this one. Does the PI have to be a clinician?
Thanks, Adam. No, the PI does not need to be a clinician. Obviously, if you're running a clinical trial, we might expect there to be clinical support to ensure that kind of patient safety, but the PI does absolutely not need to be a clinician. Approximately about 1/3 of our currently awarded portfolio are projects led by non-clinical academics.
And back to a NERG question for you, Sophie. NERG eligibility, substantial grant income defined as grants or fellowship 3 years in duration with more than 50k direct science cost, excluding PI salary per year. How do you define direct science cost? Is it consumables or also salary of a technician, shipment costs, etc.?
I don't know if I know the answer to that. What direct science costs, if there's a definition on the NERG pages or not.
I can jump in for that one.
Yeah.
We are currently looking at how that kind of cap in terms of previous awards is being how it's being defined within across all of MRC, but it typically tends to refer to that 50k cost tends to refer to consumable costs specifically every year for every grant. But we are, as I say, that is looking to be refreshed and potentially changed as we move forward.
Thanks, Helen. So I'm going to go back to the one that we didn't answer earlier and take the privilege of answering it myself. So will the NERG scheme be extended to the DPFS scheme too? So we are having discussions within our wider sort of translation strategy about how we support early career researchers across all our schemes. With DPFS, one of the first things we say is these aren't research grants, these are product development grants. So we're not 100% sure that the current criteria around NERGs would necessarily translate straight across. We are trialing it with experimental medicine just to see how it goes in these more clinically focused, often team science related grants, to see if they do work. The idea would be that we do have something for DPFS at some point, which is around supporting those early translational researchers. Whether it's a NERG scheme or not is up for debate. But what we want to do is make sure we get the right scheme to support the right people to have the right careers within translational science. So at the moment, no, but it's something that we are in wide discussions of. We've got one more question here. Sophie, Does it need to be a clinical population? How about studying healthy participants to study a mechanism that would apply to clinically relevant scenarios?
No, it doesn't need to be a clinical population, and as long as you can justify why studying the mechanism in healthy participants will answer your hypothesis, then yeah, we do fund studies in healthy participants.
Thank you very much. I'm just going to give anybody 30 more seconds to put in any more Q&A, but we are about 4 minutes from our 1 hour. I'd like to thank Helen and Sophie for a fantastic presentation and answering everybody's questions. I hope you've all found it useful. It was fantastic to see that there was, I think, 66 people on the webinar at its peak. I hope you've all found it useful, and we hope that it translates into some really interesting experimental medicine proposals over the next couple of years. So thank you all for coming today. If there's nothing else, then we'll close down this webinar.